Hello and welcome to Nature Nano Talks. I'm Fabio Plizzi, I'm Chief Editor for Nature Nanotechnology, and I have the great pleasure to talk today to Markita Landry from UC Berkeley. So hello Markita, and thank you very much for talking to us today.
All right, thank you for having me.
Yeah, sure. And today we're talking about CRISPR Cas, or CRISPR to make it simpler. And CRISPR is probably undeniably is one of the most popular invention or discoveries in science in the last 2 decades at least. And it's incredible for its potential because of its simplicity, if you like, and also its versatility. And among the application it has been really investigated for, agriculture is certainly one of the prominent ones. But here we're talking about something slightly different. We're not talking necessarily about just applying CRISPR to plants and to crops, but using nanoparticles with CRISPR in combination with CRISPR. So why is that? What do you think? Why do you think that's important at all?
Yeah, I think for most, if not all, CRISPR applications, a new tool is great, but then being able to use that tool is a completely different ballgame. So, for example, when it comes to CRISPR, there are many different ways that CRISPR can be implemented to genetically edit living systems. All of that starts with being able to get molecules that code for CRISPR or CRISPR itself from outside to inside the cell. And that's easy for certain organisms. That's kind of medium difficult for others. And I would say in agriculture for plants, it's extremely hard within this range of ease of getting these molecules in. So where nanotechnology comes in is in the sense that nanotech for really the lifetime of its existence has been a really nice player with biodelivery because the sizes of synthetic nanoparticles are very similar to the sizes of pores and membranes in biological systems. And as such, these nanoparticles can help facilitate the delivery of genetic cargoes, including those for CRISPR, into biological organisms, especially the ones that are currently the most difficult to genetically transform.
So what is in principle, you think that, let's say, there's certainly an advantage, at least from a fundamental perspective of using nanotechnology, but in many ways, does it mean that it's easy to do that, or are there challenges in using nanotechnology as well?
Yeah, of course. So I think initially it's important to consider what it is that we want to accomplish. So if we again consider the example of plant cell versus most other cell types, what are some of the barriers that need to be overcome? So here, what I'm showing is a schematic of a plant cell. Most cells and most organisms, the dominant barrier that needs to be considered for delivery is this lipid membrane. So this is a squishy lipid bilayer that is somewhat amenable to delivery of these biomolecules with carriers or through its disruption using techniques such as electroporation or heat shocking. So this is a squishy membrane. It's quite selective as evolution would have it be. However, there are tools that enable delivery in these systems. If you consider now the plant cell wall, plant cells have this porous but very thick cell wall that provides structure to plants. So plants don't have a skeleton, yet they still need to be able to grow upright. And part of what provides that rigidity, in addition to the turgor pressure of plant cells, is this highly thick and selective cell wall. And if we just do a back of the envelope calculation, the particles that can enter plant cells where the main membrane or the main barrier is this lipid bilayer are on the order of 500 or so nanometers. To get things into plant cells, the plant cell wall pores are on the order of 5, 10, 15, maybe 20 nanometers, but no more. So nanotechnology comes into play to be able to cross these membranes in a way that that is very difficult to do for plants specifically. Again, because the plant cell wall membrane is quite a unique barrier in plants. Now, where nanotechnology, I would say, needs further advances in these types of approaches is in the compatibility of nanotechnology with biological, not just cells and tissues, but also a lot of the buffers and the high salt, perhaps high sugar condition, high protein content conditions that plant cells and plant cell culture is currently undertaken in. So the colloidal stability of nanoparticles being kind of the bread and butter of the nanotechnology community, and the compatibility of the technologies that we are developing to be used in conjunction with existing buffers and existing plant cell culture workflows. There's still, I would say, a significant gap there, and it's often not clear to nanotechnology specialists what it is, what conditions are needed for the translation of their technologies from these in vitro conditions to in vivo conditions. And conversely, it's often not clear from the plant biotechnology community that colloidal stability of nanoparticles and nanotechnology is not something to be necessarily taken for granted across a broad range of conditions. So I think this is an area where the two communities would need to come together to enable further developments in this space.
In any way, this means that there is a lot of room there for working together.
Absolutely.
But there is also some other type of challenge that I wanted to ask your opinion about, and it's the fact that, if we think about genetic engineering in general, is certainly a field that requires careful regulation, both in terms of safety, but also in terms of what the public think about it. Then you take nanotechnology in itself, and that as well has all this problem related to safety, regulation, and public perception. Now you're combining the two. It seems almost like just simply combining also the problems and making it insurmountable. So what do you think it's a situation? If we can you envisage that we ever going to get there, or using the two combined, considering all the problems that are beyond the scientific obstacles themselves?
Yeah, I think as scientists, we have not done a very good job of communicating the technologies that we are developing, and that goes back several decades when the first transgenic plants were brought to market, the flavor saver tomato, which is a tomato that can be transported across longer distances due to its genetic modification, and the regulatory, or the way that regulatory oversight has gone over these crops. So currently, the way that we would, for example, generate transgenic crops is through the delivery of genetic material using one of these two technologies here. So either agrobacterium mediated delivery, which is a pathogen that can transfect plant cells by the delivery of a specific gene vector that would be engineered into the agrobacterium, or through ballistic particle delivery, so using a gun to just force these genetic materials into plant cells. These are the technologies that are currently used for the genetic modification of plants. They work well, but both are subject to regulatory oversight of the transformed crop as a genetically modified organism. And that's here in the United States, and that's also in many places abroad. So as an example, these are some of the currently approved genetically modified crops that are produced in the United States today. And some of these crops are intended for human consumption. Others are intended for use as animal feed or biofuel feedstocks or other purposes. And certain crops, a nice international example I like to highlight here is the production of golden rice, which is a variety of rice that has had the biosynthetic pathway for vitamin A engineered into it. And the concept here being that this strain of rice could be produced and distributed to populations that suffer from vitamin A deficiency. All of this kind of taken together, we're still spending a lot of time and a lot of money, over 10 years, $140 million or so, per genetically modified crop that's produced. And that is due to the time it takes to work with plants and the lack of tools to, again, get things into plant cells. That's also due to the regulatory burden of how these transformed crops are then both accepted by the public and regulated by regulatory bodies. In the United States, at least, there are currently 2 figures of merit that constitute a genetically modified organism to be intended for human consumption. The first of which is that there is trace transgenic DNA in the plant genome, so integration that happens through the use of these tools. And the second of which is if that plant tissue was generated via exposure to a pathogen. So even agrobacterium, while it's highly efficient, and even though there are ways of then segregating or crossing out transgenes from the transformed crop after its manipulation, just the use of a virus or a bacterial pathogen still elicits regulatory oversight over that crop. And so there's a burden there. Not to say that there shouldn't be. However, it's important to also consider what the alternatives are, right? We need genetically modified crops to sustain our global population, to be able to produce these crops as our land mass decreases, as climate change becomes more stringent. So in the absence of better ideas or better technologies, this is really going to be the way forward if we don't want to get to the point where, thinking back to Haber-Bosch and nitrogen fixation, where we're really at the point where we can no longer sustain our global population. And I think this is perhaps where nanotechnology can come in to provide abiotic tools that would be perhaps a little bit more compatible with regulatory oversight while still being used in the lab, and also dissemination of these tools, explaining how they work to the public.
Well, okay, that seems to be well, it's extremely exciting and challenging at the same time. But I think we can also safely say that we're still at the beginning, really, of this field, and it's what makes it, in many ways, so exciting as well. Well, I'd like to thank you again, Markita, and by the way, I would like to remind everybody that Markita is among the co-authors of a recent perspective that was published in Nature Nanotechnology, and if you want to look at the details, please go and check the perspective out. But for the time being, I'd like to thank you again, Markita, and thank you for being with us. And best of luck with everything.
Thank you very much, Fabio.